So, welcome to the second webinar in Tecan's 2013 series, a lesson for cell signaling studies using advanced detection techniques. I'm Ashley, I'm the Managing Editor for Technology Networks, and I'll be moderating today's event. The webinar will present clear and concise information on how to optimize the performance and get better results from your plate reader. For example, it'll be shown how to select the most suitable excitation and emission wavelength there, what influence the bandwidth has on the signal. Furthermore, for cell-based assays, the optimal read function discussed and how this improves results for live cell-based assays. The webinar also focuses on fluorescence detection and to help researchers fine-tuning the plate reader settings for optimal performance. The webinar forms part of Tecan Academy, an ongoing commitment and offering by Tecan for end users. The presentation will be given by Thomas Hengskill, who's an application specialist for Tecan. Just a few general housekeeping rules as well. So following the webinar, we will have a Q&A session, and we would welcome any questions that you may have. To ask a question, you should click the Ask a Question button to the right of your screen, enter your email address and ask a question, and click OK, and we'll try to answer as many questions as possible during the time available. If you experience any technical issues, please use this button to request support. So I will now hand over to Thomas.
Thank you, Ashley, for introducing me. As mentioned, I'm Tom. I work as an application specialist for Tecan, and today I'm here with the second webinar of Tecan's webinar series, and our topic is the optimization of signal-to-noise or blanking ratios in fluorescence. As scientists, we spend a lot of time to select the best assay for our experiments to get the best result. However, it's equally important to also select the right settings on your multimode reader for the selected assay to get even better results. Today, our main focus will lie on fluorescence assays. Fluorescence is the major use technology in life science research. It is worth to mention that today's topic will cover all multimode readers. So this is not a Tecan specific webinar, but this content is relevant for pretty much everybody having a fluorescence capable multimode reader in his whole lab. After the webinar, you will have learned how to optimize your cell-based or biochemical assays by either 14 or even 21-fold without ever touching the assay and just tweaking the settings of your multimode reader. So I have selected a rather simple experimental setup in order to be capable to follow all the steps we do, and that we don't have to focus too much on the assay itself. So what I'm using is 1 nanomolar fluorescein dissolved in a 0.01 molar sodium hydroxide, blank or buffer. So what are the settings we will look today? The first one is obviously the excitation and emission wavelength, which is the most important setting for fluorescence. Next, we will look at the difference between reading a plate from the top and from the bottom. And for plates which we read from the top, we will explain the effect of set optimization or set positioning. Then we will look at the effect of multiple reads or the number of flashes. And last but not least, especially for cell-based assays, we've implemented a feature called optimal read. But let's start with the most important one, the wavelength. When I started the experiment, I asked myself, what wavelength should I use? And first, I went to the vendor homepage where I bought the fluorescein from. They suggested an excitation wavelength of 460 nanometers and an emission wavelength of 515. As an alternative, I also went to the homepage of fluorescence microscope producer, and they recommended rather different wavelengths with the excitation at 493 and the emission at 517. So leaving everything the same and just entering the wavelengths, so using the standard settings suggested by the software, I first used settings or the central wavelengths from the fluorescence vendor. What I found is that with those settings, we have equally high signal as we have blank values, which results in a signal-to-noise ratio of 1.9. For the measurement settings from the fluorescence microscope producer, we get over values. Over are another rare scenario for fluorescence measurements, since this stands for oversaturation. It is fairly important for the fluorescence measurement to understand why we get oversaturation. So we obviously have to invest some work in the wavelengths to optimize the wavelengths to get either a lower background or to get rid of the over. Over, as mentioned, stands for oversaturation and is caused by an overlap of the excitation and the emission bandwidth, as shown on the illustration on the right side. So the bandwidth for the excitation and also for the emission can be found either in the software or if you have a filter device on the filter themselves. Therefore, the selection of the appropriate measurement wavelength also depends on the bandwidth you use. We can prevent this overlap, as shown in the animation on the right side, by moving our measurement wavelengths apart. Since this is a very important topic, we will have a closer look at it on the next slide. So how do we now select the right measurement wavelength? To avoid a spectral overlap, we have to define a so-called minimum distance between the excitation and the emission wavelength. This minimum distance highly depends on the device you're using. Therefore, it is essential to understand or to learn more about the technical details of your device. If we want to keep it to a minimum, all we need to know is the excitation bandwidth, the emission bandwidth, and in this case also so-called safety factor. So if we look at the minimum distance of the M200, we have a fixed excitation bandwidth of 9 nanometers, a fixed emission bandwidth of 20 nanometers, a recommended safety factor of 5 nanometers, yielding a minimum distance of 34 nanometers. For filter devices, as mentioned, the bandwidth exclusively depends on the filters you're using. So, if we now look at the new wavelengths, we have the excitation at 490 nanometers and the emission at 525 nanometers. This is 1 nanometer more than we would actually be required to use, but it's fine as long as it is bigger than the minimum distance. And generally, we should stick to the minimum distance, as this will most of the time yield the highest sensitivity and the highest signal. Performing now a fluorescence measurement using our new wavelengths, we find that we not only have a higher total signal, but also a comparably reduced background, which yields a signal-to-noise ratio of 1.5. But since the background is still comparably high, we have to look into reasons for the high background. Just on a side note, as you saw in the previous slides, fluorescence spectra are enormously important for the fluorescence optimization process. So fluorescence spectra can either be recorded with a monochromator or so-called scanning device, or can be found at various internet resources. So, as mentioned, we have to find a solution for the high background. Our previous experiments were all performed in transparent plates, where we ended up with a signal-to-noise ratio of 1.5. For white plates, we already get a slightly better signal-to-noise ratio, but in general, we highly recommend using black plates, or for cell-based assays, black plates with a transparent bottom. And using black plates for the assay results in a signal-to-noise ratio of 9.0, based on a reduced background. Another reason for high background can be the media, the autofluorescence of the media. We have tested the most commonly used cell culture media for autofluorescence at our specific wavelengths. And what we found is that phenol red and 1% FCSS results in a substantial high background. There is also RPMI without phenol red and FCS available. The next parameter we will look at is the so-called set optimization. Since different assays require to have different sample volumes in your microplate, and knowing that the sensitivity also depends on the filling volume in your microplate, it is essential to have a set optimization, or if you want to, a height control implemented in your reader. As you can see in the video, the reader is searching the surface of your sample. Coming in with the signal-to-noise ratio of 9 from our previous optimization step, we now can achieve the full optimization of 21 using this set optimization feature. This feature is for biochemical assays or for cell-based assays where cells are in suspension and not adherent to the bottom. And for bottom adherent cells, we offer the option to read your plate from the bottom and not from the top. So using GFP transfected cells, which are adherent to the bottom, we now, instead of measuring the plate from the top, we measured the well from the bottom. And this yields a signal-to-noise optimization of 14. Another option to not increase the signal-to-noise ratio, but to reduce the CV, the coefficient of variation, which is similar to the standard deviation, measure of the precision of your assay, is to increase the number of flashes. In this experiment, we have tested different flash numbers in terms of their coefficient of variation. And what you can see from the chart is the more flashes you use, the smaller your coefficient of variation will be. Or, in other words, the higher your precision will be. We recommend using flash numbers between 32 and 50. Please keep in mind, the more flashes you use, the longer your measurement will take. Last but not least, we also implemented a special feature for cell-based assays where cells adhere to the bottom, which is called optimal read. If we are doing cell-based assays with non-confluent cell numbers, we do run into the problem that the machine might read the micro well at a point where there are no cells. To prevent this problem, we have implemented a feature that is reading the well at multiple points. As seen in the animation, the machine reducing your chance that you measure at a point where there are no cells and you end up with the wrong signal. At the end, this will contribute also to your signal-to-noise ratio. So from the experiment where we used human squamous epithelial carcinoma cells, which have been stably transfected with enhanced green fluorescence protein, we found that independent of the cell number, whenever we use optimal read, we have an increased signal-to-noise ratio. So to sum up our findings, well, we started with a rather suboptimal signal-to-noise ratio of 1.1. We now, tweaking all the features of our multimode reader, were able to boost the signal-to-noise ratio of a cell-based assay to 14 with a significantly higher signal and lower background, and we were even able to boost it to 21 for biochemical assays. We achieved that by optimizing the wavelengths using a fluorescence spectra, changing from transparent to black plates, which we recommend for fluorescence, and to solid black plates for biochemical assays, and black plates with a transparent bottom provided by Greiner for cell-based assays. The set optimization, which optimizes the distance between the sample and the detector, and the mode where we read the plate either from the top or from the bottom. As promised in the beginning, we optimized the signal-to-noise ratio for cell-based assays to 14 and for biochemical assays and suspension cells to 21. If you would like to learn more about fluorescence measurements or also optimizing other technologies, please go to tecan.com/academy, where we provide high-quality courses for detection technologies. Please follow us for the next webinar in the Tecan webinar series. Thank you and goodbye.
Well, thank you very much, Thomas. I'm sure you'll agree. That was very, very interesting. So we've had a few questions come in during the period of the webinar. I wonder if I'll put these to you, if you'll be able to answer these for me. So the first question we've had in is optimal read a better choice compared to normal well scanning?
Well, Andrew, you see, optimal read gives you the opportunity that the device is actually still measuring at multiple points in your well and is then forming the average. So at the end of your measurement, you will only get one single value instead of multiple values. So this basically simplifies your data analysis a lot.
Okay, brilliant. Thank you for that. The second question we've had in is, what gain function should I use if I have the same sample volume in every microplate well?
That's a good one, since we didn't even cover gain in the webinar, but I suppose you still saw it on the screenshot from our software. The point is, if you know your assay and you know at which position you have the highest concentration of your fluorophore, this might be, for instance, from a standard curve, you should choose the well with the highest concentration. If you don't know which well has the highest concentration, just choose optimal gain, and the device will select the gain for you.
Okay, excellent. Thank you for that. And the final question we've had in is, can I really do multiplexing with a Tecan plate reader? And is it easy to combine absorbance and fluorescence measurements, which are common setups, obviously, for long-term cell-based assays?
That's a little more Tecan specific, this question. That's, since I mentioned at the beginning that this is a general webinar, but here I can only speak for our software.
Okay.
Control. With our software, you just have to pull in the measurement strips. So we provide stripes for absorbance, fluorescence, time resolved fluorescence, and so on. And all you have to do is grab one of those stripes and pull it into your workspace if you want to. So depending on how many measurements or different measurement modes you would like to use, just pull in the strap.
Okay, excellent. Well, thank you very much, Thomas. That covers all the questions I've had in. So I'd like to thank you again for your time and for your excellent presentation. And really just want to remind people that this webinar is going to be available on demand. You'll receive notification of when that will be. So that's obviously one that you can pass around to your colleagues as well who maybe didn't have an opportunity to attend. And I'd just like to take this opportunity to remind people of the next in the series of the Tecan Academy webinars, which is coming up on September the 27th. So another date for your diary there. And obviously we'll send you more details near the time. So thank you very much for attending.